Always, I think let's start. So,
Okay, ma'am.
welcome everybody for this exciting session on molecular testing basics to automation. So, without any delay, I will hand over to Oves. And Dr. Oves, I will log out because I've made you and Dr. Pratna co-host. So, please take it forward. Thank you.
Okay. Thank you so much, ma'am. Thank you, ma'am, for giving me this opportunity. And thank you, IAMM. Good evening, everyone. Today's topic is molecular testing from basics to automation. And this lecture is a continuation of our ongoing series on automation in microbiology. So far, we have covered automation in bacteriology by Dr. Sonal Saxena, ma'am, and serology by Dr. Sanjeev Gogoi. In this session, mm, we will cover what molecular tests are and their principles behind them, how they work, and their role in diagnostics, and how automation has transformed these techniques. We'll start with the fundamentals of molecular methods used in microbiology and then their evolution to the fully automated system. Now, unlike some other areas of microbiology, automation in molecular testing is not just an add-on or a supplementary tool. It has now become an essential part of the process itself. Let's begin by asking, what do we primarily do in microbiology? We do detection. Is the organism present? And we do identification. What is that organism? And we do characterization, determining features such as antimicrobial susceptibility, etc. The same things we can achieve by detecting the DNA or RNA of the organisms. Because all the organisms, they contain DNA or RNA, and molecular methods, they have an advantage over phenotypic methods because every organism has DNA or RNA, and it does not matter whether the organism is fastidious, hard to grow in lab or not, or how much time does it take to grow. It is slow growing or fast growing. Molecular tests, they bypass all these issues and give us faster and more direct results. And molecular methods, they are also capable of characterizing and identifying organisms better than phenotypic methods. For example, if you are doing a microbiome study, all the organisms will not be able to grow in the culture, and there will be difficulty in identifying closely related organisms by phenotypic methods. So you won't get a true picture of the bacterial population in the microbiome, whereas the molecular method will give you the complete picture of it. And then there is also a crucial issue of biosafety. While for culturing, you might need level 3 or level 4 containment. In contrast, for molecular testing, once you have inactivated the sample, the rest of the process, the downstream process, remains the same. You inactivate the sample either by adding the lysis buffer or some other methods, then it is the same method for all the organisms. So this is another advantage of using the molecular methods. Now I will begin with some basic properties of the DNA, RNA. DNA is an information molecule, and sorry, it replicates. With the help of so many enzymes, it replicates inside the cell. And this happens in vivo, inside the cell. In vitro, the DNA, on increasing the temperature, it denatures. And on decreasing the temperature, the two strands of the DNA, they again come closer and they bind to each other. That is called annealing. And this annealing or this hybridization is sequence specific. It happens only when the complementary sequence is found on the other strand of the DNA. Besides this, if you add enzyme, polymerase enzyme, DNTPs, etc., this replication of the DNA can also happen in vitro. This all is basis of all the molecular tests that we perform, and we'll see how it does it. We have primarily two types of, broadly speaking, we have two main types of molecular testing method. First, we have a non-amplification method or hybridization method, in which if we want to detect the DNA of a pathogen, we use a complementary DNA sequence that is called a probe, which is labeled with a reporter molecule. If the pathogen's DNA is present in the sample, the probe binds to it, and we detect the signal from the reporter. In another method, that is we called amplification based method, we take advantage of the fact that we can replicate the DNA in vitro by adding enzyme and other things, and we can multiply the target DNA many times until it becomes detectable by the machines or by some fluorescent dyes. This technique is called target amplification, and this method is called non-amplification technique or hybridization method. Hybridization, sorry, hybridization technique is direct. You can compare hybridization technique with microscopy, where you can see whatever is there. It gives you a snapshot of what is present as it is. Whereas the target amplification, it is similar to culture in microbiology. You are increasing the number of copies of the DNA, just like you do in culture. You increase the copy of the bacteria so much so that it becomes visible. But there is an important point. Hybridization methods can only work when there is already sufficient amount of DNA, target DNA present in the sample. If the DNA is very low, it might not be detected by the probe, because our capture mechanisms, cameras, they can't detect a single molecule. You need to have at least 10 to the power 11 molecules or something. So we have a third category called signal amplification, in which we do not amplify the target DNA. Instead, we increase the intensity of the signal. That is called signal amplification. So to summarize, we have three types of molecular detection method: hybridization techniques, no amplification, then signal amplification, and the target amplification. Now, one more key aspect, regardless of the method, we need access to the pathogen DNA. The DNA must be available outside the cell, which means we need to first extract it, not just extract it, but purify it. Maybe for hybridization, you can use the extracted DNA, but for amplification techniques where you use many enzymes, you need purified DNA. DNA extraction and purification is often most time consuming and limiting step. So let's begin with the DNA extraction. Nucleic acid extraction. There are three key steps in the nucleic acid extraction. One is disruption of the cell, then digestion of the protein, and purification of the nucleic acid. Cell disruption can be achieved by physical disruption of the cell or chemical disruption of the cell. Physical disruption by mechanical method, ultrasonic, pressure, temperature, osmotic, or chemical disruption of the cell by organic solvents, alcohol, chloroform, chelating agents, EDTA, detergents like SDS, tritone, or by enzymes. Once you have disrupted the cell, then next step is the digestion of the proteins that can be achieved by enzyme proteinase K. Then we need to purify the nucleic acid, because even after this, all things will be present in the lysate. Proteins, digested proteins, and all part of the cell membranes, etc. So we need to purify the nucleic acid out of this lysate. And it is based on one property of the nucleic acid, that it precipitates in presence of alcohol and salt, and it dissolves in water. So on the basis of this property of DNA, we can separate DNA from all these impurities. There are two methods: liquid-based extraction and solid-based extraction. Liquid-based extraction is the classical method of DNA extraction. It is phenol chloroform extraction. We add phenol chloroform in equal quantity, 50-50 ratio. And what happens, the basic principle is the organic solvent base. Different macromolecules, they have different solubilities in these solvents. So phenol will disrupt the proteins, Chloroform improves the phase separation and enhance the protein denaturation. And aqueous buffer, it holds the DNA. So in this phase, after this, we either centrifuge the tube or we keep it for a long time. Then there will be three phases. One is aqueous phase, interphase, and organic phase. The upper aqueous phase will be containing DNA or RNA. The interphase will have proteins and cell debris, and the lowermost will have phenol chloroform plus the lipids, etc. Then you can take this upper part and you can precipitate the DNA with alcohol and salt and separate it. The advantage is it gives a very high yield and long fragments of the DNA. And RNA can also be separated. You add trizol in it so that the pH of this aqueous part becomes less than 4. So only RNA comes in this aqueous phase and DNA remains in the interphase. So with liquid phase extraction, you can also extract RNA. And it is cost effective, no need for kits. But disadvantages are there are toxic chemicals involved, it is labor intensive, and it is not friendly with automation. And there is emulsion formation, and there can be carryover of the inhibitors. This phenol and chloroform can inhibit our downstream applications like PCR and sequencing. And of course, it is time consuming. So we commonly use solid phase extractions. Solid phase extractions are based on the property that DNA can bind to a solid surface or it can precipitate on a solid surface like silica in presence of alcohol and salt solution. So the most common solid phase method is silica membrane based extraction method, in which we have a double tube, the inner tube and an outer tube. We add absolute alcohol in the lysate so that the DNA get precipitated and it binds to the silica membrane present in the inner tube. And then we centrifuge it. When we centrifuge it, the DNA remains bind to the silica membrane and the lysate comes down into the outer tube. And after 1 or 2 washing steps, we can add elution buffer. DNA again becomes soluble and it comes into the elution. And then we centrifuge it again. The DNA along with elution buffer comes into the outer tube. This is silica column based DNA extraction. We can achieve the same thing by using magnetic beads coated with silica or polystyrene. And these magnetic beads, again, in presence of alcohol and salt, can bind with the DNA. DNA will precipitate along with them. Then we apply magnetic field. DNA along with magnetic beads come to the wall of the tube. Then we discard the supernetting, that is with all the impurities. Then after washing, we add elution buffer. And when we add the elution buffer, DNA again becomes soluble. Then we apply magnetic field, all the beads come to the wall of the tube, and the DNA, we decant the DNA, purified DNA. This is called magnetic based extraction. This is called spin column based extraction. The magnetic bead based extraction is easy to automate. So nowadays, most of the systems which use automated nucleic acid extraction, they use magnetic bead based extraction. For example, this is Roche MagnaPure system. This is MGI system. This is Thermo Fisher Kingfisher Flex. They all are magnetic bead based DNA extraction system. They are just liquid handler along with a magnetic plate. So principle in all of them is same. I'm not going to compare them in this lecture, but we should take care in mind whenever we are going to choose automated extraction system. There are few points that we need to keep in mind. One is the throughput. It refers to how many samples the machine can process in one run. It can be 8, 16, 96, etc. Now, it may seem like larger number is always better. If your lab works on that much of sample, then larger number, higher number is okay. But if that is not true and you do an occasional testing, get few odd samples, then high throughput systems might not be cost effective, and you can use a low throughput system. Another thing is flexibility. Which type, whether the machine can handle different type of samples like fluid, swab, stool, tissue, etc. Some systems are optimized only for liquid samples and may require significant pre-processing, like stool sample or biopsy sample. Then level of automation. What is the level of automation? Some machines are fully automated. They just you just load the sample and the machine does rest of the job by using preloaded cartridge. While other machines, you will have to manually load the reagents into it, the samples into it, and this fully automated systems are better because semi-automated and there are more chances of error, risk of error, and it is also time consuming. Next is protocol versatility. It is important to know, can you customize the protocol or can you use a third party reagents? Because some machines, some systems are closed, meaning they only work with their own branded kits, not the outside kits. Then fifth is downstream compatibility. Some machines can integrate with the PCR system or the sequencing system, or at least they can help you make the master mix plate in case of PCR, or they can help you make the library for sequencing. So this all should be kept in mind. And some machines are equipped with barcode tracking and sample ID system that is important for diagnostic samples. Now, it is not that you can't automate the spin column based extraction. Their limiting factor is centrifugation, because centrifuge is very difficult to integrate with an automated system. So there are systems based on spin column extraction, but they do not use centrifuge. Instead, they use either vacuum or positive pressure to move the lysate through the silica membrane. One example is Kaizen Cube, Egg Cube, or Take on Freedom. These are spin column based automated extractors. So this was about the DNA or RNA extraction. Let's begin with the hybridization techniques. As I mentioned earlier, DNA has a key property that it denatures at 94 degrees centigrade, 94 to 98 degrees centigrade, and it anneals at 50 to 60 degrees centigrade, but only with a complementary sequence. We use this principle in the molecular diagnosis. Hybridization can be solid phase hybridization and liquid phase hybridization. In practice, solid phase hybridization is more common. So solid phase hybridization can be, we first fix the DNA or on the nitrocellulose or nylon membrane. It can be of four types: dot slot hybridization, sandwich hybridization, southern blot hybridization, and northern blot hybridization. Dot slot hybridization or dot blot hybridization, both terms are used. In this technique, it's very simple technique. First, we denature the DNA and then bind this DNA to a membrane. It can be a nitrocellulose membrane or polystyrene membrane. Then this membrane is immersed in a solution containing the probe. After hybridization, we wash away the excess probe and we detect whether the probe is bound to the target or not. For example, in this case, it is a fluorescent molecule, and if we will apply UV light, it will give fluorescence. And if the fluorescence is there, it means the probe has bound to the target DNA. It means target DNA was present in the sample. So the advantage is you can test many samples for many targets on a single membrane, because the membrane itself is plain. Example, this is a single membrane, and you can test in this. They have tested up to 42 samples for these many targets for tuberculosis. So on one single membrane, they have done so many tests. On 42 samples, these many targets have been used. This is dot slot hybridization. Another variation of hybridization is sandwich hybridization. In this, all the process is same as dot slot hybridization, but the DNA, the solid surface is already bound with a capture probe. The capture probe binds to the target DNA from the sample. Then the rest of the process is same. You will immerse it into the hybridization probe solution. Then you will look for the hybridization. The advantage is that you do not need UV cross linking or heat for attaching your DNA to the solid phase. You will buy this nitrocellulose membrane along with this already bound capture probe. And another advantage is because there are two probes, obviously the specificity of the test will increase, right? And in this sandwich method or other method, I have shown here a fluorescent molecule, but it does not have to be necessarily a fluorescent dye. It can be another system of detection. It can be a labeled probe with biotin or deoxygenin, which can bind to an enzyme labeled antibody or HRP conjugate or enzyme conjugate. This enzyme conjugate can give you a colorimetry reaction that can be detected, or it can also give you a light generating reaction with luminol or H2O2 like chemiluminescence. So both the techniques can be used for sandwich hybridization, colorimetric and chemiluminescence. The example of the colorimetric technique is BD-AFORM VP3 assay. This machine uses the colorimetric method of sandwich hybridization, and it detects simultaneously 3 targets on Gardnerella vaginalis, Trichomonas vaginalis, and Candida species. The example of this chemiluminescent hybridization is histoplasma capsulatum detection. This is made by AccuProbe. It is just like how we do it in immunological testing. It can be colorimetric or chemiluminescence, fluorescent, or radioimmunoassay, although we do not use radioactive substance nowadays. Next important thing is this is southern blot hybridization. This is another classical thing of the hybridization. In this, we either extract the genomic DNA or do the amplification. Then we do the gel electrophoresis. We separate different size of DNA. Then we transfer this DNA onto the nitrocellulose membrane, and then we apply a probe. Here it is a radioactive probe. It does not need to be a radioactive probe always. It can be fluorescent probe or any other probe. Radioactive probes are not used nowadays. And then it can be detected by X-ray film because this is a radioactive in this way. This is called southern blot hybridization. Another variation of it is northern blot, in which we use RNA. It was discovered by Edwin Southern. Northern blot hybridization, the technique is same, but it is for the detection of RNA. Yes, this all was solid phase hybridization, where the DNA was either the DNA or the probe was bound to a solid phase. Now, another method is solution phase hybridization, in which all this process is happening in a tube, in a solution. But how will we wash away the excess probe in it? The answer is we use S1 nuclease digestion. This is a exonuclease, which digests any single stranded DNA. So if the probe is bound to the DNA, it will remain there. Any DNA which is single stranded will get digested. So then we can look for, by precipitation, we can look for the double stranded DNA in the sample. This is liquid phase hybridization. So in all these methods of hybridization, the first requirement is DNA extraction. But when we do the DNA extraction, we do not know about the cells. What were the cells present in the samples? So there is another modification of it is in situ hybridization. Instead of extracting the DNA from the tissue, we perform hybridization directly on the tissue, maybe the histopathology slide. We prepare a thin smear of this tissue, and after a series of preparation, we remove the paraffin, we digest with protease enzyme to reveal the nucleic acid targets, and then we fix it with formaldehyde, and we apply the probe directly on the histopathology slide. The advantage is that you can know which cell has the target DNA and which cell does not have the target DNA, as opposed to your extraction, post-extraction hybridization, in which it will just a solution of everything. Here also, on the basis of your detection molecule, reporter molecule, if it is a fluorescent molecule, this is fluorescent in situ hybridization. And if it is chromogenic molecule, it is chromogenic in situ hybridization. This is called CISH, this is called FISH. Here we are using enzyme conjugated probe and colorimetric substance, just like we have seen before. Here we are using fluorescent molecule labeled probes. So FISH and CISH are two methods of in situ hybridization. And this is an example, the in situ hybridization. Here it is cancer cells are there, a cervical cancer tissue sample tested for HPV DNA using FISH. Some cells show bright fluorescence indicating presence of HPV. Other cells have no signals, that is, HPV is not present in the cell. So this cannot be achieved by hybridization after extraction. So this is the advantage of FISH, that you can see not just whether the target is present or not, but also where it is present. The location of the target DNA can be detected by FISH. One modification of FISH is PNA FISH. Usually hybridization is done on or by the RNA molecule or DNA molecule. This is ribonucleic acid. This is deoxyribonucleic acid. There is another big molecule, natural molecule, that is peptide chain. We can create an artificial molecule by attaching nucleic bases to this peptide chain. This thing is called peptide nucleic acid. This is ribonucleic acid, deoxyribonucleic acid. This one is peptide nucleic acid. It is an artificial molecule, DNA bases attached to a peptide chain. Now, the thing is that this peptide chain can hybridize to the DNA. And this hybridization is again sequence specific, and it is stronger. We can detect it with the help of a fluorescent molecule, and it binds stronger and more specific way than the DNA probe. Another advantage is that because this is an artificial compound, it is resistant to nucleases and proteases. So there is less degradation inside the cell. And because it is a smaller molecule, it gives better resolution. And it is more stable at higher temperature in harsh conditions. And one big advantage is it can cross the cell membrane, whether it is transporter molecule attached to it. You can attach a fluorescent molecule in one end, another, and you can attach a transporter molecule, and it can enter into the cell, even inside a live cell, living cell. So you can, this is the advantage of PNA FISH. This technique is called PNA FISH, peptide nucleic acid fluorescent in situ hybridization. So advantages, you can use it, suppose in blood culture positive bottles, you can make a gram stain smear, and after gram stain smear, you can make a PNA FISH smear. And this will give you fluorescence depending upon the bacteria, whether it is present or not, or any other target you can choose, like MACA gene for MRSA. This one is PNA FISH. You see the intact cell is giving fluorescence. This is green one is Candida albicans. This red one is Candida glabrata. This is from a positive blood culture bottle. And in this, you can see both of them. And this, none of the target is present. Another example is, this is from, again, from positive blood culture bottle. And this is Staphylococcus aureus. If you are using a probe specific to Staphylococcus aureus, it will just tell you it is Staphylococcus aureus or not. If you are using the probe specific for MACA gene, it can tell you whether it is MRSA or not, right? But one thing is important before doing PNA FISH, again, gram staining is must, because this will prevent unnecessary use of the PNA fish. It will help you choose the right probe. Otherwise, you will be using MRSA probe in E. coli if you do not do the gram staining of the bottle. And now coming to microarray, it is an automated solid state hybridization technique. It can detect thousands of targets at once. It consists of a solid surface, usually a glass slide, and thousands of DNA probes are fixed on a grid-like pattern. And when we add the labeled sample, DNA or RNA, and apply it to this chip, and then we wash it, then we look for the fluorescence. This is the chip, a part of the chip. It is attached with probes. And if we add the sample and sample is complementary to these probes, it will bind along with the fluorescent molecule. And this small area is part of a bigger area on the chip. And like this, there are thousands or hundred thousands of probes attached to this whole chip. And this is part of this bigger glass slide. And this can be detected by laser. So the advantage is it can detect multiple targets in one test, and it can be used for pathogen detection and identification, amplification, antimicrobial resistance profiling, mutation detection, virulence factors detection, and gene expression profiling, comparative genomics and strain typing, and microbiome analysis. Also, you can do the 16S profiling of the whole sample, because it is detecting so many targets at the same time. And it can also detect threat agents. So this was hybridization technique. Now coming to the signal amplification technique. Signal amplification is achieved, classically it is achieved by branched DNA. But as we have talked before, our in solid phase hybridization, that enzyme-based methods, they are also a kind of signal amplification, because the enzyme, because it is not taking part in the reaction, it is just one molecule, but it is capable of producing more than one molecule, whether it is colorimetric or chemiluminescence. So that is also a signal amplification. But classically, signal amplification is achieved by a special probe that is called branch DNA probe, or bDNA probe, where the one DNA is attached with small DNA probes, and this way the signal is enhanced. Each branch of the DNA has one reporter molecule. By this way, no target amplification is there, and it can be quantitative. It can detect even a smaller quantity of DNA. The advantage is there is no risk of contamination. There is no risk of amplicons generation, and no thermocycler is needed. That is the advantage of this method. These are some pictures showing the branch DNA probes. Another signal amplification method is T2 magnetic resonance method. In this, it uses magnetic nanoparticles coated with captured probe. These are the target DNA, and these are magnetic nanoparticles coated with captured probe. There are two type of nanoparticles, two types of probe. One will bind to one end, other will bind to the other end. And if the target DNA is present in the sample, so they form, by binding to each other, they form a cluster, and it alters the relaxation time of water molecules. That is the principle of magnetic resonance, also used in MRI imaging. It alters the relaxation time of the water molecule present in the sample, and this signal is captured by the machine. And this is a fully automated machine. It does not require DNA extraction. It does not require amplification, although inside the machine there is some kind of amplification that is proprietary technique, but it does not require outside. It does not require amplification or DNA extraction, and it is highly sensitive, can detect as low as 1 CFU in whole blood. The major limitation is the panel is restricted. For example, one panel has only 5 targets: Enterococcus, Staphylococcus aureus, Klebsiella pneumoniae, Pseudomonas aeruginosa. These are the common organisms for sepsis, because this is mainly used for uh, the diagnosis of sepsis. Another panel has all 5 species of yeast: Candida albicans, Tropicalis, Cruzi, Calabrata, Parapsilosis. And this is, as I said, it is completely automated. You just add the vacutainer in it, not even your blood culture bottle, just the vacutainer, without culture, without amplification, without extraction. Everything is processed inside the machine. That is automated hybridization. Now coming to the uh, target amplification. PCR target amplification, or PCR, which is sometimes considered synonymous with molecular techniques. But here, this is the DNA replication, which happens naturally. It requires a lot of enzymes, helicase, which opens the 2 strands of DNA, polymerase enzyme, which extends the single-stranded DNA into double-stranded DNA. Then there is ligase, which will connect the nicks of the DNA, and topoisomerase, which reduces the tension of the DNA. This happens in vivo, but in vitro, we achieve all these things just by altering the temperature and giving the reaction only one enzyme, that is DNA polymerase. And with each cycle of heating and cooling, these double-stranded DNA will separate, and it will become 2 strand. They will polymerize, and in one cycle, we will have 2 DNA fragments. And like this, after 30 cycles, we can generate a lot of DNA. This is what I think everyone knows, but I want to know this is because of the properties of the DNA. It's annealing and denaturation, but we also should know the property of the DNA polymerase enzyme to understand it correctly. The DNA polymerase enzyme, one property is that it replicates only a double-stranded DNA. It can't convert a single-stranded DNA into double-stranded DNA. It can only extend a small double-stranded fragment and make it a fully double-stranded fragment. This is one property of the DNA polymerase enzyme. And another is that it has a 3 to 5 prime exonuclease activity. If a wrong nucleotide is bound to the fragment, it will go back and remove it. This is 3 to 5 prime exonuclease activity. Another property is 5 to 3 prime exonuclease activity. If while it is polymerizing this strand and on the way it finds a small fragment of the DNA, it will just remove this fragment. It will cut this fragment from uh, and it will synthesize its own DNA fragment. It's not like it will come here and will bind the 2 fragments. It will first remove this and then synthesize this DNA. So it has this 5 to 3 prime exonuclease activity, 3 to 5 prime exonuclease activity, and it requires a double-stranded fragment, which we provide by providing it a primer. Naturally, the primer is produced by the primase enzyme, but in PCR reaction, this primer is provided by us. And this gives us the ability to amplify a specific part of the DNA. We can dictate to the DNA polymerase that we want amplification from this area to this area by providing the primers. If DNA could amplify without this, we could not have done the PCR. So these properties are important to know to have the better understanding of the PCR reaction. Then you require the template DNTPs. This is the template DNA which you have extracted. Then you require DNA polymerase, DNTPs, buffer, cofactor, and the primers. These things together nowadays called master mix. In research or older days, people used to use all these things separately, but in diagnostic labs nowadays, This together comes as a master mix. You just add two things in the master mix, DNA template and the primer, and you can go for the PCR reaction or the amplification. This is how it is done. But this is not enough. There are more to it. PCR was discovered by Karim Mullis. He was awarded Nobel Prize for that. And the steps of PCR, as I said, there are three steps: extraction, then denaturation, annealing, and extension. This is achieved by rapidly changing the temperature of the tube. Earlier, it was done by water bath with three segments in it for different temperatures, one for denaturation, other for annealing, other for extraction. But nowadays, and in each cycle, they earlier we used to add polymerase enzyme, because polymerase enzyme will denature in denaturation process. So nowadays we use a thermostable polymerase, or the Taq polymerase acquired from Thermus Aquaticus, which is thermostable. And instead of that water bath, we use a machine with a metal plate, and which can rapidly cool and heat itself. This is called thermocycler. So thermocycler itself is an automation, I guess. And with the help of thermostable polymerase, you can just put the tube, one single tube on one single site, and after some time, the complete amplification will be processed. You don't have to change the tube for different temperatures. So there are various type of thermocyclers. Some has this plate system, others have a capillary system, and the hot and cold air is blown on that capillaries. They are much faster because heating this plate and cooling this plate takes a longer time. But capillary based systems are faster. They do the PCR in less time. There are some variants of the PCR. One is multiplex PCR, in which you multiple sets of primer to amplify multiple targets in one reaction, or nested PCR, 2 rounds of PCR to increase the specificity. And the second PCR uses primer inside the amplicon of the first primer. So the advantage is it is extremely sensitive and as well as specific. Once you have done the PCR, next step comes to detect the PCR product, because even after completion of the PCR reaction, the tube looks still the same. Earlier it was transparent water. Now also it is transparent water. You don't know whether the PCR amplification has happened or not, or the target has been amplified or not. So we have different methods for the detection of the PCR product. One is gel electrophoresis, very commonly used, or we can use some dyes which bind to the double-stranded DNA, or we can do probe-based detection. This is the gel electrophoresis, the conventional method. Separate PCR product by size on the agarose gel. We add the DNA here, then we apply the current, and the DNA is separated on the basis of its size. And we can visualize it with the help of dye. Can be ethidium bromide or cyber green under UV light. You can take a picture, and in the picture you will see the band. The positive sample will give you a band of the size which you would already know. This is a classical method of detection of PCR product, and this is end point detection, because you are detecting the PCR product at the end of the test. Another method of detection of PCR product is CRISPR based method. CRISPR is an enzyme based system in which there is an enzyme called Cas9 and there is a guide RNA. This guide RNA binds to the Cas9, and whenever it finds the sequence complementary to this guide RNA, it breaks the DNA from that point, both the strands of the DNA. So this is CRISPR-Cas system. It is used by the bacteria for the protection from viruses or some unknown sequences. Whenever they find that sequence, they break that sequence. So in diagnostics, this can be used for at many places, but here we are talking about the end point detection of the amplicon. Once the PCR is completed, we can use this Cas9 system, and when it binds to the target DNA, it gets activated and it breaks the probe, which has a reporter molecule and a quencher molecule. And once the probe is cleaved, it will get detected. This is CRISPR based detection. This is again a endpoint detection. Another modification of this CRISPR based detection is Cas9 editor linked uniform detection assay. This is FAM Cas9 editor. F is for FAM, also known as Feluda. It was developed in India. In this Cas9 is not used to cut the DNA, instead to bind to the DNA. When this guide RNA binds the target sequence, it binds to it, and this target sequence, with the help of biotin, bind to the test band line, and this FAM molecule attached to this guide RNA uh, is captured by the antibody coated with gold particle. So just like all other rapid immunochromatographic tests, this this amplified product is detected by the antibody and gold particle. So this is Feluda. Uh, Again, I would say this is a detection method because all the previous steps are already there. It was used for the COVID-19 detection during the outbreak, but it does not eliminate the requirement of the DNA extraction. It does not eliminate the requirement of the PCR. It is just a method of detection of the PCR product. It just eliminates the requirement of your real-time PCR machine, which detects the PCR product in real time. So if you do not have a real time PCR machine, you have just conventional PCR machine, you can use this method. So time taken, say it is said that it takes only half an hour. I don't know how that half an hour is counted from here only, from PCR step or from extraction step, right? This is Feluda. It was developed in India. There are previously it has been already used. This one was already used, but the mechanism is slightly different. Here it is breaking the probe. Here it is just helping binding the guiding RNA to the target probe. If it will not bind to this, this will get washed away and it will not give the band. So again, these all methods are endpoint detection methods, be it gel electrophoresis, Feluda, or turbidity detection, or fluorescent endpoint detection like qubit. They have a disadvantage. It takes a longer time, and you know everything only at the end of the reaction. And also you have to open the tube. There is again chances of contamination, amplicon contamination. So to overcome these disadvantages, we have another method, that is real-time PCR. It detects the PCR product as it is being amplified. That's why it is called real time. It uses fluorescent dyes or probes that emit signal proportion to the DNA amount in each cycle. As the PCR amplification process progresses, the amount of DNA will increase, so the amount of fluorescence will also increase. This is the normal PCR constituents of the PCR reaction. We add one probe in it. This probe has a quencher molecule and a fluorescent molecule. When it binds to the DNA, and when PCR cycle goes on, the DNA polymerase finds it on the way and it breaks it by the 5 to 3' exonuclease activity. And this probe gets separated from the quencher molecule and it gives fluorescence. With each passing cycle, the fluorescence will increase because one probe with each passing cycle will get degraded. Or same can be achieved by adding a dye which binds to the double-stranded DNA, because after each cycle, the concentration of double-stranded DNA in the tube will increase. So this can be detected by a camera for the fluorescence, and it gives a curve. Right, this is the curve of fluorescence, and this curve can help you in the quantification of the target molecule also, because there are some components of this curve. One is this is the baseline phase of the amplification, where amplification is there, but it is not able to detected by the fluorescence. After some cycles, it starts giving fluorescence, which is detected. Now, at what cycle it crosses the baseline, that is called CT value. If the target in the initial tube is more, the CT value will be less. The cycle number where the fluorescence crosses a predefined threshold indicates the detectable DNA. The lower the CT value, the higher the initial amount of the target DNA. So by this way, we can do the quantification by comparing the CT values to a standard curve or the controls. You can estimate the initial quantity of the target DNA in the sample. The advantage is it is faster. It can do the quantification. It can reduce the contamination risk because you are not doing any downstreaming gel electrophoresis. You are not opening the tube, and it is more specific and sensitive because other than primers, you have one more thing to make it more specific. That is the probe. At least in the real-time assays, which uses the probe, it will not be true for I think cyber-green real-time PCR. The uses of it can be used for gene expression profiling, how much a gene is expressing, viral titer determination, quantitative pathogen detection, species identification, or syndromic identification of pathogen by multiplex PCR. Yes, that is another thing. You can use multiplexing easily in real-time PCR by using different fluorescent dyes for different targets. These are two methods of detection method. One is non-specific binding, cyber green, which non-specifically bind to the double-stranded DNA. Other is the fluorescent probe. This is the picture showing how the fluorescent probe works. The tech polymerase, it cuts the probe into two and releases the fluorescence. There are other types of probe. One is Fred probe, where there are two probes and they bind adjacent to each other. One is quencher, one is excitation. This molecule is excited and there is emission of light with this. Another probe is molecular beacon. This is a folded probe. They are all used in real-time PCR. There are some PCR-free methods, amplification methods like LAMP, loop-mediated isothermal amplification. This is commonly used. And in this, there are multiple primers, overlapping primers. And when the amplification cycle goes on, the strand, amplifying strand removes the primer from the, and in each cycle, um, of the amplification, although there are no temperature cycle, but at the same temperature, this amplification goes on again and again because the extending strand removes the primer from the side. Now, there are some fully integrated, as I said, PCR itself is an automated system, but there are some systems which can integrate the whole process, starting from the DNA extraction, PCR amplification, and detection. These are fully integrated sample to result PCR systems. One is GeneXpert, developed by Cepheid. It is cartridge based, minimal hands-on time. It is GeneXpert. It is cartridge based systems, and you just load the sample in the cartridge. It is a sealed cartridge, and it automates the nucleic acid extraction, amplification, and detection inside the cartridge. And there is less time taken, and there is no, or at least minimum, biomedical waste generation in it. Another system is Roche Cobas 6800. It was used in during COVID time in many centers in India. It is very high throughput, fully automated system. It can process 100 to 1,000 samples per day, and it was extensively used during the COVID time. And it also integrates the sample preparation, PCR amplification, and detection seamlessly. Other machines are BDMax system or the Panther Fusion system. They all are fully integrated platform for doing all the processes one by one on the same sample. Another example is the TrueNAT. it was developed by Mole Biodiagnostics. It is a Goa-based company, and it is also a fully automated system, but it is not so compact because DNA extraction occurs in a separate machine. Then you take the extracted DNA and put it into the another machine, and it is available as 1, 2, or 4 chip ports. It is approved by WHO and ICMR, and it is used in national TB program for the rifampicin resistance, as well as for the detection of tuberculosis. It is battery operated, so very suitable for remote clinics, mobile diagnostic units, and primary healthcare centers. It was used. It was also used for COVID-19. So a little bit about the sequencing. It is also an important aspect of molecular testing. Sequencing is not so much used for the patient care, for the diagnostic test in microbiology, although it is used for research and etc. And all the process, they are automated systems. I will just briefly talk about the principle behind those techniques. This is one base pair of nucleic acid. This is the ribose sugar, and this is the phosphate group, and this is the nitrogenous base. And depending on this nitrogenous base, there will be 4 type of nucleotides. And what happens? These are the 4 type of nucleotides. They bind each other. This is the 5' end of the sugar. This is the 3' end of the sugar, and each 5' will bind to the 3' end of the sugar. And this way, the DNA gets amplified. That 5 to 3, 5 to 3, 5 to 3. What happens if instead of these regular nucleotides, if we can add a blocker molecule on this 3' side? So what will happen? Suppose this is the amplification of the DNA will stop at the site where this blocker nucleotide will bind, because there will be no free 3' end. So these blocker molecule, these nucleotides are called D-deoxyribonucleotides. Wherever they bind to the chain, they block the amplification of the DNA. So suppose this is the normal PCR, we add polymerase enzyme, nucleotides, and we get the amplification. Suppose in the same reaction, we add dideoxynucleic acid, adenine, in a small amount. So what will happen? Instead of getting this complete clean band of same size as amplicons, we will get these type of amplicons. They will be of different sizes, because wherever this dideoxyadenine will bind, the cycle will stop then and there. So instead of getting this, we will get this. And if we do same reaction in 4 different tubes by adding 4 different nucleotide, so we will get another picture. And here we know that at the end of each fragment, there is adenine. And if we repeat this for all 4 molecules, 4 nucleotides in 4 tubes, we will get this picture. And with the help of computer, or with the help of, earlier they used to do gel electrophoresis, and they will combine all these fragments. And this, if we combine all these fragments, they will be seamlessly, with the difference of 1 nucleotide, will give the whole picture. There will be fragments of every size with the difference of 1 nucleotide, and you would know that this has come from that tube, this has come from that tube. By this way, you can know the complete sequence of the DNA. This method can be automated by using, instead of proper gel electrophoresis, you can do the gel electrophoresis in some capillary tubes, and you can use 4 different fluorescent molecules. With the help of that, you can automate it. This method is called chain termination method, or Sanger shotgun sequencing. Because the chain is being terminated, this is chain termination method. This is the first generation sequencing method. In the next generation, what we do, instead of having a blocker molecule attached to these nucleotides, we can add fluorescent molecule. And not just one type of fluorescent molecule, we can add 4 different kind of fluorescent molecule on 4 nucleotides. And these fluorescent molecules, they are reversible. You can attach it. Once it is bound to the DNA, you can remove this fluorescent molecule, and the 3' end will be free again to bind to another nucleotide. So in that case, we will add first one type of nucleotide. If it is bind to, if it is bound, it will give a separate, different kind of fluorescence. G is bound, so suppose it gives green color, and then blue color, or we then remove the fluorescent molecule, we again proceed for the cycle. So suppose in next cycle thymine is bound, so it will give red color. So like this, the camera will pick the color with each cycle, and it will give the sequence of the, this is called sequencing by synthesis, because we are able to remove the blocking agent from the nucleotide, and that blocking agent is nothing but the fluorescent molecule. And that fluorescent molecule is four different color fluorescent molecule. This technique, when attached to a solid surface, because the camera will not be able to capture fluorescence of one molecule, you need to first amplify the product, and you first amplify the product on a solid surface, and then you do the same sequencing by synthesis. This technique is used by Illumina sequencing. This is called second generation sequencing or next generation sequencing, or the proper name is massive parallel sequencing. Now, another thing is there is a phosphate group attached to this molecule. Whenever nucleotide binds to the another molecule, one phosphate group is freed, and from this end, one H ion is freed. So from 5' end, one pyrophosphate will be freed, and from 3' end, one H ion will be freed. And if you use this pyrophosphate molecule, and it will form the ATP, and with the help of luciferase enzyme, it gives light. That is used for in the pyrosequencing. So instead of fluorescence, with each molecule added, you can detect the light. That is used by the pyrosequencing method. Another way is of the detecting of the H ion that is being released by every cycle. And this H ion is really captured by the change in the pH, that is ion torrent. That technology is used by ion torrent semiconductor sequencing. This is second generation sequencing again. Another is the third generation sequencing, in that we try to sequence longer molecules and sometimes single molecules without amplification. One method is the nanopore sequencing, in which the DNA is passed through a pore present in the membrane. And while this DNA is passing through the pore, the electric current between the two side of the pore is changed. It depends on the which nucleotide is passing at that moment, A, T, G, C. There will be different change in the color of the uh, uh, in the change in the resistance of this hole. So this technology is used by Oxford nanopore sequencing. In this, we can sequence longer fragments, and without amplification, we can use this. So these were some basic aspects of sequencing. I just wanted to cover it just for the sake of completion. Otherwise, sequencing will take, I guess, another lecture, 1 or 2 lectures. That's all from my end. If there are some questions, I would like to take those questions. The one question is from Rohit, is asking, what is specific there in elution buffer? Elution buffer, sometimes it is just distilled water. Sometimes there are some buffers are there, but even distilled water can do the work of elution buffer. Although the kits do not tell you what is there in the elution buffer, but many a times it is just distilled water. or they can add some buffer just for the stability of the DNA. How to get access to the old lectures on YouTube? You just type on the YouTube IAMM Masterclass. You subscribe to it. There is a channel, and all the lectures are present on it. I'm not aware of any WhatsApp group for this masterclass. PPTs, I don't think are being shared, but these lectures are present on YouTube, so you can just go and watch the lectures. Ananya Chakrabarti is asking best source to study this from. Study what? You mean molecular testing? You can read it from, I would suggest ASM and different for newer techniques, because in ASM you won't find newer techniques or their examples. You can look for certain papers. I'm very sorry, I wanted to add in the end these references, but I forgot, sorry. But you can read ASM. Some chapters are there in all your microbiology books regarding molecular testing, but I personally find the ASM better for it because it includes almost everything, and research articles. What is the principle of magnetic beads? Magnetic beads, Rajat, where you are asking it in the extraction or you are asking in T2MR? Because both the places there was magnetic beads. Okay, let me answer first this. How do I use ASM? Any advice for DNB student? Use ASM? Actually, when we used to read in our PG, we used to read different things from different books. From ASM, we used to read virology and molecular, etc. For bacteriology, we used to read Ropley. It depends. Let me answer this question of Rajat about the magnetic beads. Magnetic beads are coated with some polystyrene or something. So when you add alcohol and salt in the DNA precipitates and bind to the magnetic beads. So this bound DNA with magnetic beads gets attached to the wall of the tube when you apply the magnetic field. So you can then wash, you can decant all impurities, then you can wash it. And when you want the DNA released from the magnetic beads, you just add elution buffer or distilled water. The DNA will again come into the solution phase. It will become soluble. It will get separated from the magnetic beads. Then you apply again magnetic field. All the magnetic beads will come again side. You can just take your DNA in the elution buffer. That is the principle of the magnetic beads in extraction. In T2MR, magnetic beads, they are coated with probes. For one sample, one target, they are coated with two probes. So the two probes will bind to one end of the target and the other end of the target. So just like antigen reaction, it will form a cluster. All the magnetic beads will combine together and form a cluster. And these clusters, they interfere with the magnetic resonance of the water molecule, which is the principle of MR. So that interference is captured. If it is there, it means target is present. If it is not there, the target is not present. That is the principle of T2MR. Okay, thank you, everyone. I guess there are no more questions. we can end this session. The question asked by someone is how to you see the frequent future classes. I think the link is always the same. Every Friday, 6 o'clock, you can just open the link, even if it is not. But it is either on the website and they through the WhatsApp they inform it. Okay, thank you so much. I'm ending the session.